قدامك اهو لما last session we spoke about plants nutrition, okay, and we explained this is autotrophic, they are autotrophic organisms, or this is autotrophic nutrition, okay, at which the plant makes its own food by process called photosynthesis. Okay, and we handle the equation of the process of photosynthesis, and we defined it as a process at which the plants take raw material like water from the soil and carbon dioxide from the air to make its own food or complex compound like glucose using energy from the sunlight, which is absorbed by the chlorophyll. And we had these two equations, word equation and symbol equation. Then we go through the adaptive feature of the leaf as an external features, right? And internal features of the leaf. And we learned how to label each part and what is the function of each part, or the role of each part for the process of photosynthesis. Then we explained the timing at which stomata will be open or closed according to the need of the leaf for exchange of, of gases and according to the change of the rate of photosynthesis. And I said respiration always going on inside the plant cell, but the photosynthesis is the one which will be varied according to the light intensity. Okay. And then we spoke about the guard cell, and I explained this diagram for you how the uptake of oxygen or uptake of, or the release of oxygen and uptake of carbon dioxide will be varied according to the daytime, and this due to the change of the light. Then I give you the fate of glucose. Glucose is the only product which is formed for the process of photosynthesis, and the plant can use it to make many products like amino acid and protein, like chlorophyll in the presence of magnesium, like protein in the presence of nitrate, make starch, make cellulose, make sucrose, lipids. Time. Factors affecting a rate of photosynthesis. Okay. We said there are three factors affecting the rate of photosynthesis: light intensity, carbon dioxide, and temperature. Okay. Who can define the term limiting factor? If you know what's limiting factor, just please send message on the chat so I can read it. What is limiting factors? What are limiting factors? No, factors affecting the rate of photosynthesis. Yes, agree. One student wrote factor affecting the rate of photosynthesis. Of course, light, carbon dioxide, temperature, all needed for the process of photosynthesis, so all affect the rate of photosynthesis. The question is, Why is it called limiting factors, not only factors? Limiting factor for any process which is controlled by more than one factor, okay? The one which found in such shortage is the limiting factor. What does it mean? It means, for example, if I have plant here, okay? This is a plant, okay? Found, or let's say it's aquatic plant, right? Okay. And I I put water inside this plant, okay? I will put that plant in water and I will put carbon dioxide, but no light. No light. I will keep it in dark. If this plant in dark, there is photosynthesis or not? Okay, no photosynthesis. Okay. Okay. So here no light, no photosynthesis. If I will plot graph here, okay, here I will write light intensity, and here I write rate of photosynthesis. Okay, zero light, zero photosynthesis. طيب. I will make an opening here in that dark box, that cardboard box. Okay. Then I will change that dark to light by putting lamp here. So this light will go to the plant, right? Here is light. Okay. So there will be photosynthesis. Agree with me or not? So more light. I put here one lamp. More photosynthesis. So the light limited the process, or the process was not occurring because the absence of light. Now there is one lamp which is giving light. Maybe not that much strong light. Okay. But there is light going to the plant. You understand? Okay. Right. There is photosynthesis. I can see if there is photosynthesis or not by collect the oxygen produced using gas syringe. Okay. I can use Benedict reagent to test the percentage of glucose formed after particular time, or whatever what we have, we can do. But there is photosynthesis. Okay, I will put one more lamp. When I put, when I put one more lamp, okay, it means what? It means more light intensity. Okay, there is one more lamp here. So more light is getting out from here, going to the plant. So here the light intensity increased or not? Increased. So there will be more photosynthesis. Agree? Hmm? I will put one more lamp here. So more and more light going to the plant. Okay? So more photosynthesis. More lamp. So the more the lamp I am putting, and pay attention, all these lamps are in the same distance from the plant, more photosynthesis till I reach to the limit at which, okay, so at that time, the more I'm putting lamp, I'm increasing the light intensity, then I reach to the limit where there is living off. What does the living off mean? It means I'm increasing the light intensity, as you can see here, okay, but no effect on the rate of photosynthesis. It means the plant don't need more photosynthesis to increase its rate. Okay? Before that, the plant needed photosynthesis because, sorry, they needed the light for photosynthesis because there was shortage of the light, right? It was limited. That's why it's called limiting factor. So from here to here, this part, I can say light is limiting factor. Okay, light is what? Limiting factor. Okay, why? Because light was found in shortage. So the more the light I will increase, more photosynthesis. Till it reaches to the limit at which that light cannot increase the effect of the rate because the plant have light more than needed, not in shortage, high light intensity. The plant may need more carbon dioxide, may more suitable temperature. So here, the reason here is that not light intensity, not limiting factor. What will be the limiting factor? Why the plant not increase in the rate now? Maybe carbon dioxide, okay, concentration need to increase, okay, maybe light, sorry, maybe temperature. You got my point? Give me that. Okay, you understand this part? Hmm? You got it? So limiting factor means the factor which limit the process. The process cannot increase without increasing that factor. You understand? So when there was no light, there was no photosynthesis. So I added one lamp, photosynthesis start to occur. I added two lamps, become faster. Three lamps, faster and faster. After that, I'm adding more lamps, no more increase in the rate. So the plant is oversaturated from the light. Give me anything else if you would like to increase the rate. You understand this part? Hmm? Okay. Jumana, you're raising your hand. You have a doubt? Type. If I would like to plot a graph, okay, for the carbon dioxide concentration and the rate of photosynthesis. Okay. Who can sketch that graph? Can you sketch it in the board? Hmm? Okay, Rama, you can. Alia. So it will be the same shape exactly like the light. Yes. Okay. Exactly correct. Thanks. So here the rate of photosynthesis was increased with the increase of the carbon dioxide till here. After that, there is level of or constant level of the rate. So more, no increase. You have any doubt about that or not? Right. From here to here, what was the limiting factor? Hmm. Yes, carbon dioxide concentration. And you have to mention the word concentration, don't say carbon dioxide only. And here, okay, not carbon dioxide. It could be light intensity or temperature. So here carbon dioxide is the limiting factor. Okay, here carbon dioxide, not limiting factor. Okay, what will be the limiting factor? Maybe temperature. So here limiting factor would be temperature or light intensity. And you have to use the word limiting factor in your explanation of this graph, okay? Because they will ask you in terms of limiting factor, explain this graph. So you will look for the limiting factor, okay, for the rate of photosynthesis at each part of the graph. You got my point? Okay. طيب, here I will sketch also the relationship. Here, temperature. Here, rate of photosynthesis. I think this graph, okay, similar to the relationship between temperature and enzyme activity. Agree with me? So I can understand from that graph the effect of temperature on the rate of photosynthesis is due to the effect of temperature on the enzyme's activity. Is it clear? Hmm? You have any doubt regarding that part? Okay, okay. Gamal, Khalid, you have doubt? If you have doubt, you can write it in the chat, please. Yeah, what is your question? Write it again, Rama. Mm, water, if it will not be uh, water, if it will not be found or limited, the plant will stop photosynthesis, you can say, and close the stomata. So it's in some question, in some answers, it could be considered as limiting factor, but يعني water have to be found because if it's not found, the plant will close the stomata and may photosynthesis will stop. Is it clear? Huh? Hmm. Any other doubts? Yeah, always we plot the limiting factor in on x-axis because this is the variable which we are changing during the experiment. So the changed variable always will be in x-axis and the results will be on y-axis. Okay? Ukuni, Rahma. Okay, no more doubts. Okay. Okay, Rama. Okay. You have any doubt? Any doubt? So we finished talking about limiting factors. Okay. In some planning on some experiment, here the way that we collect the oxygen produced by displacement of water. Okay. Right. So the oxygen will be collected above the water. And sometimes we are using what we call the heat filter, like this one. This is like measuring cylinder or long jar. at which we will put water. Why we will use that as a heat filter? You know, water high specific heat will not allow the heat to pass, okay, to reach the plant. So it prevent or work as like barrier for the heat which getting out from the plant and heat up the heat which is getting up out of the lamp to heat up the plants. You got it? You have no doubt about that? And this is the part about limiting factor. This is another method at which we can collect the volume of gas produced using gas syringe. And here, this tube have to be tightly closed. Okay, to prevent any gas leakage. So all the gas will be collected here in the gas syringe, and I can measure the volume of gas produced within unit time. And this will help me to calculate the rate of the reaction. تمام? And this is the effect of temperature, as we said. You have any doubt about that part? Hmm? This was the last part for the plants nutrition. Any doubt in that chapter? Hmm? If you have any doubt any doubt regarding to any part of that chapter, just write the question in the chat, Zoom chat. Okay, no doubts. Type. I just wanted to go through the practical part of that chapter. Okay. How can I give approval that chlorophyll is needed for the process of photosynthesis? How can I approve that light is needed? Okay. How can we prove that carbon dioxide is needed? All of this related to the practical. First of all, you have to know that once the plant make the photosynthesis, some of the glucose will be stored in the chloroplasts. يعني chloroplasts store starch. So if I will test the presence of the starch and it's found in the chloroplast or in the leaf, it means there was photosynthesis. If I tested the leaf for presence of starch and it was negative, I used iodine solution and it was negative, it means, okay, word equation and symbol. You have a doubt about that part? Hmm? Do you hear me? If you hear me, write yes in the chat. Okay, thank you, Ramah. Okay, so I can make distortion of the leaf. يعني I remove the starch from the leaf by simply putting the plant in a dark place, putting the plant in dark pot. Okay, sorry, dark cupboard or dark cabinet. So no photosynthesis. So the plant will use the stored food, which is the starch in the chloroplast. This is the meaning of distortion of the leaf. تمام. I can take the leaf and make iodine test directly. Iodine with starch gives blue-black color, but the chlorophyll, if the leaf will stay in that color, that chlorophyll color will mask the blue-black color, which will be formed due to the iodine test with the starch. Especially, it's a very, very small granules and chloroplast inside the cell. So we have to decolorize the leaf. Here is the steps to decolorize the leaf by putting the leaf in boiling water. Then we'll take the leaf from boiling water, okay, to put it in ethanol. يعني hot ethanol, but not heating it. Then we will take that ethanol and put it back again in cold water, right? We're going to put the leaf in hot water to break down the cell membrane so easier for the chlorophyll to be extracted. And I'm putting it in ethanol so ethanol can extract the chlorophyll. And هنا put it back in water one more time. يعني this is water, okay? To the fragile, easy to be broken. So now when I put it in water, it will not be fragile, so we can use it. Okay, the leaf ready now for the iodine test, so we can test it and we can see blue-black color if there is a starch. Okay. If I would like to make an experiment to give approval that light is needed for the process of photosynthesis, okay, here I will select one leaf, okay, from a plant and cover one part with a black paper strip or dark thick paper, so it will not allow this part to absorb light here. Okay, so here no light absorption. No light absorption means no photosynthesis. Then I will make the starch test on the leaf. So this part are blue-black, this part are not. So this is mean light is needed for the process of photosynthesis. You got my point? Okay. Here, this is a variegated leaf. This one, it's inverted like that. Parts of the leaf have chlorophyll, parts of the leaf don't have chlorophyll. Okay. So here we will make the starch test directly instead of putting the board or the paper on the leaf. Then I will keep it in the sun for particular times. Then I come to see the test. This, if I have plant which have this type of variegated leaves, that I will do the test directly. Of course, the parts which are green will do photosynthesis. The parts which is not green will not do photosynthesis. So the blue-black parts will be for the chlorophyll parts, and the other will not have the result. Okay, this is approval for the test of importance of chlorophyll for the photosynthesis. تمام. Here carbon dioxide is necessary or not. Again, I will take two plants, same age, same height, same leaf, or the same plant even, but I will do the experiment twice. Here I will put sodium hydroxide solution. Sodium hydroxide solution absorbs the carbon dioxide. So here the surrounding of the plant without carbon dioxide. Sodium hydrogen carbonate releases carbon dioxide. So here there is carbon dioxide, here no carbon dioxide. Then I will take the leaf, okay, from each experiment, okay. Then I make the starch test. طبعا these two plants have to be de-starched before the experiment to be sure that there is no starch. You got my point? To measure the rate for the experiment of limiting factors, this one of the experiment I show you in the drawing. I will use the plant, and here is heat filter, and here is lamp, right? Heat filter here, which helps to maintain the temperature of the experiment, because also when you're doing the experiment of limiting factors in plants, I have to be sure that only one factor is changed, not all factors. Yani if I'm measuring the effect of light intensity, I have to be sure that temperature is maintained constant. I have to be sure that the carbon dioxide concentration is constant. No, any other factors can affect the rate except the light intensity. Is it clear? Okay. Here, always we have to keep the lamp at particular distance from the plant. If I increase the distance, the light intensity will decrease. If I increase the distance, if I increase the distance, the light intensity will decrease. If I decrease the distance, the light intensity will be increased. You got it? Here's the other method by collecting the volume of gas produced using gas syringe. Another method also, he will use meniscus and capillary. Here, the oxygen will be produced, pressure will be built up, and this bubble of air will start to move towards the air. Okay. Here, oxygen will be out as waste gas, right? And I can measure the distance moved by this air bubble to measure the rate of the reaction. Okay. Another method here, we can take discs from the plant and put it here, and we'll try to adjust the leaf to be found in the bottom of the cup like that. Okay, just no need to raise the stem and put lamp. So this leaf disc will do photosynthesis. I can, when the oxygen will be formed due to photosynthesis, this bubble of oxygen will make the leaf to float up. So I will measure the time taken for these leaves to float up to the surface of the cup. You got my point? يعني I will measure time taken for this leaf to float, okay, from here to here. You understand? By this way, I can measure the rate of photosynthesis. You got it? Hmm? This all about the practical part of this chapter. Okay. I think by that way this chapter is done. These are the features of the leaf external, and here is the internal one. We have to memorize the function of each part. Okay. Next session, inshallah, we will start in the plants transportation. Okay. That plant nutrition is done. Are you okay with the plant nutrition? If you would like to me to stop explaining and would like to start revision sessions for chapters that you need, it's totally fine. But tell me which chapter you are struggling, or you need more than the others. Because some of you asked me to explain plants. That's why I made plants nutrition. Then I will go to plants transportation. Plants nutrition. Okay, after finishing plant transportation, let's see what is the next chapter that you need. Okay, okay. Uh, Nice to see you. Okay.